Oops, sorry. Okay. Yeah, so there are structural, enzymes, hormones, transport, storage. So albumin is an example of a storage protein. Contractile. This is what you usually are thinking of. People are usually thinking of when they think of proteins, is that it like builds muscle. So muscle proteins are actually proteins, and they can contract when you are lifting something or doing something that requires that. So your muscles are all made up of proteins, which is why you need to eat protein after you exercise, because you need to rebuild it and maintain it. And then they can also be antibodies, and they can fight infection. And that's a pretty relevant one right now. So here's an image of some antibodies. So in this image, you've got antigens as viruses and bacteria. Do you guys want me to go back? Sorry. I just kind of went really fast through that slide. So viruses and bacteria, we will refer to as antigens. They're like foreign things that enter your body and you don't want them to be there. Antibodies are proteins that are going to find and attack those specific things and get rid of them. So proteins, antibodies, they help us. Have you guys heard about like the COVID antibody test? Yeah, so when they're testing for antibodies, they're actually testing for a protein in your blood that's specific to attacking and destroying COVID. Kind of cool. Be useful to have. That's also what when you naturally get a virus or when you get a vaccine, you're building up antibody proteins that are specific to that virus in your bloodstream, so that in the future your body recognizes it and makes those antibodies and fights it off. There's an image of it. All right, amino acids. So these are the monomers of proteins. Can you guys all see that molecule on the bottom pretty clearly? Mm-hmm. Okay, so carbon and hydrogen and oxygen, organic or inorganic? Organic, yeah. So amino acids are organic. And the bonds within those, so we've got hydrogens, we've got carbons, oxygen and nitrogen. Those are all nonmetals. So inside of an amino acid, holding it together, are covalent bonds. We will talk about ionic bonds holding different amino acids together, but within an amino acid itself, the bonds are covalent. And then I'm going to show you guys a picture which shows what the amine group is on the next slide and what the R group is. I'm going to show you this larger one, and I can go back if you guys need to write more notes. But it references the amino group and the R group on the last slide. So the amino group is that left side, the nitrogen bonded to two hydrogens and to the carbon in the middle. The R group, which is sometimes called the side chain or the functional group, is what makes every amino acid different. That's what the amine group and the R group are if we refer to those. Yep. No, that's okay. I mean, if you want to, but as long as you know what the R group and the amino group are. Okay. So it's the structure. There's 20 of them. So they're kind of small and hard to see. But the R group, the functional group, is what actually gives each amino acid its unique identification. So there's 20 different amino acids. And here's a few of them kind of zoomed in to show their structure. So you guys can see, you can see the amine group, you can see the carboxyl group, and you can see the R group, which is the highlighted one on all of this. And that is what really varies in the amino acids. If you ever decide to go to medical school, you have to actually memorize what all 20 of these look like for the MCAT, for the medical admissions test. Yeah. So those are our groups. Oh, and then I was just going to say there are, I don't have all of the amino acids memorized. There's 9 essential amino acids that you guys need to get in your diet. You might have heard of a couple of these actually. So histidine, isoleucine, leucine, lysine, you don't need to know this. Like you don't need to know this for a test or anything. Methionine, phenylalanine. I struggle with that one. Threonine, tryptophan. Have you guys heard of tryptophan? No. And valine. Tryptophan is one that people will often blame for being tired after Thanksgiving dinner because they're like, there's so much tryptophan in turkey that it makes you tired. There's tryptophan in all meat, but people just tend to overeat on Thanksgiving, so they get tired because they're too full, and they blame that amino acid. Tryptophan never did anything wrong. Poor tryptophan. Yeah, so those 9 essential amino acids, you have to get those from your diet because your body can't actually make those. So if you aren't eating foods that have all of those 9 amino acids in them, then you're not going to get enough of them, and you're not going to be able to do certain things in your body, like make certain proteins. And amino acids are actually something that your body doesn't store for later, so you have to get all of those kind of almost every day type thing. Whereas the other ones your body can actually make as long as it's got all right. So deamination, this is the removal of an amine group from an amino acid. So getting rid of that nitrogen side. And that image is just an example. You guys don't need to memorize that that's what it looks like. You just need to know the definition. All right, so why do we do that? We break that down into parts to use in our body. So we can get carbon dioxide and water out of it. Glucose, which we use for energy. And then the amine group actually is toxic to our cells, ammonia, and we get rid of that in our urine. So in our case, that turns into urea. In different animals, they actually get rid of that ammonia in different compounds. Like birds really concentrate it into, what's the chemical called? Uric acid. I'm not 100% sure. But that white stuff that's in bird poop is actually the ammonia portion of an amino acid, just really concentrated into this chemical. Yeah, this one? Mm. I'm going to Google it. I forget what that one is. Bird urine ammonia. Yeah, it is uric acid. So yeah, instead of urea, birds extra concentrate it. Okay, am I good to move? Awesome. Okay, peptide bonds. So these are bonds that are formed by dehydration synthesis, which is the making or breaking of a making. Awesome. So it joins amino acids together. As you can see in this image at the bottom, it's getting rid of that water, dehydration. So a peptide bond is just the specific bond that occurs when you join amino acids. And it is a covalent bond. So yeah, a peptide bond is kind of a specific version or a specific type of covalent bond. Right. And so this is our amino acids being joined together. We get rid of the water, the OH from the one on our left side in this case, and the hydrogen from the one on the right side there are where the bond is going to happen. So that O and those two H's form water, they leave, and a bond forms between that nitrogen and that carbon that are left. So that's our peptide bond. kind of cool that some of the water that's in your body comes out of chemical reactions, and you've actually made, not drank. Eh?
Very cool.
Yeah. So peptides are a really long polymer of amino acids. And we call them proteins if they have a function in the body. So dipeptide has 2 amino acids, and then when many join together, they're called polypeptide. And if that polypeptide does something in the body, we call it a protein. And this terminology stuff is going to come up in our digestive system a fair amount. Most of the stuff that we're doing right now will come up in our digestive system again. And I guess the urinary system as well, probably. So a few examples of proteins in your body. Insulin, you guys have probably heard of. It is 51 amino acids long. Human growth hormone has 191 amino acids in it, and they're all folded up. You guys don't need to know how long these are, but just know that they're examples. Hemoglobin, 574 amino acids. Crazy. And hemoglobin is all like linked up and going through your blood carrying oxygen. So packed a lot of amino acids in there. These also say protein chains. So there are amino acids in long chains. And the protein chains are separate, but then they'll be joined together. So it's one protein, but there might be a few different kind of steps to making it and then putting it together. We're halfway in some of the slides or pictures. All right, so you don't need to write this one down. This is just to show what I'm going to talk about. So protein structure. First of all, you have got an amino acid chain. We call that the primary structure. And then depending on how those amino acids interact with each other, so those R groups that we saw give each amino acid different properties. So some of them like love water and some of them hate water. So then they're going to form secondary traits where they start to fold in different ways. Like the ones that don't like water are going to go into the middle, and the ones that like water are going to go outside. And just gradually, they're going to make different formations based on those traits that they have in the amino acid sequence. So there's 4 kind of levels of protein structure that we'll talk about. And this is the stuff you need to write down. Okay. So the primary structure of a protein is just a straight chain of amino acids. If you guys thought, like, if you attached a bunch of kindergartners together on a rope and you made them all hold onto the rope, and then you put them in a room, and there were like some puppies in one place, like some Play-Doh in another place, and like some killer wasps in another corner, do you think they'd stay in a straight line for very long? No. They'd like be all over the place, like they'd be all folded up in this room. So proteins are kind of like that. Each amino acid is different, has different traits. It's surrounded all of a sudden by water and by other amino acids. So it doesn't stay in that straight line formation for very long. It starts to fold and interact with its surroundings really quite quickly. But it does, like kindergartners holding onto a rope, stay attached to itself, to each other. Some of our amino acids are polar molecules. So we've got uneven sharing of electrons. Those are going to start to interact with each other and form hydrogen bonds. There's a lot of stuff going on in protein folding. So the secondary structure starts to form because of hydrogen bonds between the amino acids. So between polar amino acids, we've got hydrogen bonding. And the secondary kind of level of structure is what makes it look kind of like a beautiful ribbon in those scientific drawings that we saw. Don't worry about knowing the terms alpha helix or beta plated sheet. That's just to show you the visual. You don't need to know those two terms. So. I'm so happy that the smoke is gone. It's the worst. Okay. Tertiary protein structure. So this is just more bonds, creating more interactions. So we've got our protein, and it starts to take on this 3D kind of globular shape. And it's held together by different bonds between more distant amino acids. So just to go back for a sec, this one, we've got hydrogen bonds happening between pretty close together ones, making a spiral. Now, amino acids that are farther away are actually starting to interact in different bonds. So we can have hydrogen bonds between some of them. We can have ionic bonds forming, some covalent bonds. And then there's one called disulfide bridges, which is if the R groups have a sulfur atom in them. Not all of the amino acids have that in their R group. Oh, okay. And you guys can see in this one, you've got that kind of spiraling happening, like inside of this, and then that's all globbing together. Our last level of structure is the quaternary whoa, protein structure. This is one of those things that I've written a lot and then rarely say out loud. Quaternary. Whatever. So multiple polypeptide chains. So this is multiple ones of these joining together. And then you get a 3D shape. And it's a globular protein. So hemoglobin was one of the examples that I gave earlier as a type of protein with a structure or with a function in your body. It actually has 4 different polypeptide chains in it. So we've got beta 1 and 2 and alpha 1 and 2. Some of our proteins just have 1 protein chain, and others have more than that.
So these are both called globular proteins?
Globular, yeah, proteins. Yeah, as soon as it has a 3D shape. And that shape really determines how it interacts with things in our body. So your body is really all about like different shapes fitting into different receptors and different chemical reactions. So a lot of different things are like, for example, with hormones, you've got receptors on different parts of your brain, different parts of your body, and they're going to be a really specific shape. So say you've got a receptor for testosterone. So you've got a receptor for testosterone, and you have testosterone in your body. And this is really simplified. This is not what testosterone looks like. But just to show you, so that would fit into the testosterone receptor really nicely. And then certain functions in your body that testosterone carries out would happen. But if you just threw some estrogen into that person, testosterone is estrogen, into that same person, and they didn't have any receptors for it, this isn't going to fit in that receptor, so it's not going to have any effect on that person. So your body is, a lot of it is just to do with shapes of different proteins and how those fit in different places in your body and chemical reactions. Okay, so back to that original diagram, we've got our amino acid chain, which is our primary hydrogen bonds from close together ones, called the secondary structure. That all bends up due to different bonds, tertiary, and then multiple chains might form together, and that's the last one. Okay, so what types of bonds actually cause that building? Like, how are these amino acids actually interacting from a distance? I think I got a bigger photo of this. In this example, in this image, there are a few different types, and it's not really easy to read on your sheet there. But it's just showing an ionic bond on the top left there, a hydrogen bond here, a disulfide bridge here, and then hydrophobic, so not water-liking, in here. And those are a few examples of bond types. I think we're going to get into bond types more tomorrow. And today, for the rest of class, actually get started on the project because you don't really need to know the bond type stuff for the project that you're doing. So we will continue on these notes tomorrow. So just kind of make sure you're leaving yourself space.